This is the final set of slides for cells. This is specifically cell communication, cell signaling. You watched the first PowerPoint in class. You should have watched 2, 3, and 4 over the weekend, and this is the last video. It is due on Friday, which is a D-Day after the trip, if you were going on the skiing snowboarding trip. So there are multiple ways of communicating between cells, and we've talked a little bit about it because we've already talked about the endocrine system, we've talked about the nervous system, and this is the most basic cell signaling. 2 yeast cells, they are unicellular prokaryotic cells, excuse me, and this is how yeast mate, because we know yeast are actually living things. Alpha cells have receptor sites for the alpha factor, and they also produce alpha factor. Alpha cells have receptor sites for the alpha factor, and when mating occurs, they are exchanged, causing the 2 cells to fuse and meiosis to occur. And so we see that not only do they have receptors for their own factor, they have it for the other, and that allows them to identify what is self and non-self cells. When mating factors are exchanged, it causes the 2 cells to fuse, meiosis occurs, and we have this combination of DNA, which makes new offspring. So these are so that is kind of the most basic type of cell signaling. These are some of the types that you need to know for the AP biology test. Local signaling. Paracrine signaling involves cells secreting chemicals that cause behaviors in other close by target cells. Excuse me. So let's say the cell releases growth factor, it causes the cells around it to multiply. Local signaling indicates that it is very close by. Literally, the cell just releases the hormone or the chemical into the extracellular fluid around it, and it reaches the cells next door. We also see that there is neuron communication. Neurons communicate using neurotransmitters, and the neurotransmitter diffuses across the synapse, and it interacts with another neuron. Again, this is local. We call it synaptic signaling specifically, but it is local, right? The cells are very close to each other. Cells can also communicate through direct contact instead of that local signaling where they are just very close. Direct contact can occur between cells that have cell junctions. We see them most often in plants with plasmodesmata or gap junctions in animal cells, and they can literally send messaging chemicals through those connections. Also, we see cell-to-cell recognition, which is one cell physically binds to the other cell and determines if it is self or non-self. Think the immune system. So those are all local signaling types of signals. We also see long-distance signaling, and this usually involves the use of a hormone. Animals release hormones into their circulatory system, so into the bloodstream, and then the hormones travel to specific target cells, which is why there is such a delayed response often. Plants release hormones that travel through their xylem and phloem, and so it can occur that way. It can also diffuse into the air as a gas, and so we see that plants use sort of like pheromones when we talk about people releasing chemicals that attract certain people to us. Plants use something like that. It's not for attraction, but it is chemicals released into the air. There are types of hormones that we need to know. Steroid hormones, for example, testosterone and estrogen, modified amino acids, which are just proteins. So thyroxine is a hormone that's modified from tyrosine, which is another protein. Insulin is a protein hormone, and it has to do with the structure. If we look at those structures below, we see that the proteins end up having nitrogen in them and things like that, whereas the steroid hormones only have oxygen, carbon, and hydrogen. You can look up the action of steroid hormones and non-steroid hormones if you'd like to on YouTube. The cell signaling stages, we've seen this before. You've seen this exact picture when we talked about the endocrine system. There are 3 stages of cell signaling: reception, transduction, and response. The activation of the cellular response that occurs inside of the cell. In non-steroid hormones, so protein hormones, Reception begins with the signal or the hormone interacting with the receptor site that's on the surface of the cell. So embedded in the plasma membrane. The non-steroid signals don't ever enter the cell. When it attaches to the receptor, it causes a change in shape of the receptor site, much like the enzyme with its activation site. And the receptors are usually proteins that are inserted into the membrane, which we've talked about. Types of membrane receptors, there are three main types that we need to know. G protein linked, tyrosine kinase, and ligand gated ion channels. Obviously, G protein are specific proteins embedded in the membrane. Tyrosine kinase we'll talk a little bit more about, and ligand gated ion channels are specific to a ligand that allows ions in and out of the cell. G protein linked receptors, they activate different things we call G proteins. They are specific to going somewhere else and causing another activity to occur. I would recommend looking up, this image does pop up if you want to see it. So our signal molecule, usually our hormone. This would be a non-steroid hormone. Binds to the G protein linked receptor. It's going to activate that yellow G protein that we see, and then it's going to activate an enzyme, which causes a pathway to occur. Tyrosine kinase receptors activate more than one signal transduction pathway. This is important when it comes to cell reproduction or things that have to have multiple pathways occur. What we see is that we need two signal molecules, and my proteins start apart. They basically come together and fuse. They use ATP, and they release multiple activated proteins to cause a cellular response. We can see it here. This process does require a large amount of ATP. We see that it loses a phosphate, making it ADP, which causes the release of energy. And then we'll see that it activates multiple proteins, the purple. The last type are ligand-gated ion channels. They are protein pores in the membrane that open or close in response to the presence of a chemical signal. They always allow the flow of ions in and out of the cell. So, for example, your sodium-potassium pump would be an example of that in the nervous system. So we have our first receptors, and then we have something called our second messengers, and it's a molecule inside the cell that triggers other things to occur inside of the cell. There are two really common second messengers, cyclic AMP or CAMP. We can see it as the orange square in the diagram, or calcium ions and IP3, also used as second messengers. CAMP is a derivative of ATP. It is an enzyme, and it's found in the plasma membrane. It converts, the enzyme converts ATP into cAMP, which becomes a second messenger, as opposed to a source of energy. And we can see that this is our G protein. We can see that the ATP, the yellow, is going to bind with the adenylate cyclase to become cyclic AMP, which is going to cause the signal transduction pathway to occur. Most of the time, calcium in the cytosol is low because it's pumped into the endoplasmic reticulum and the mitochondria. It's also pumped outside of the cell. When calcium floods into the cytosol, they can be used as a second messenger. We see this in muscle contractions. If we look at this image, what is important is we have a G protein linked receptor. It is the purple receptor. The other example is your tyrosine kinase, but we can see that in both cases, they are heading towards IP3, which is a second messenger, which is activated by different pathways, but as it moves towards the endoplasmic reticulum, which is the blue organ, it's actually going to open a calcium gated or an IP3 gated channel. It's going to open up the channel, and calcium is going to be released from the endoplasmic reticulum, and it's going to activate various proteins. Once we have that second messenger activate proteins, the cell needs to process that signal, and the biochemical pathway, or called a transduction pathway, kind of results in a cascading or a domino effect, which ends up amplifying each product. So we get a lot of whatever product we are trying to make. In the image to the left, we see that we probably have a G protein linked receptor. We see that we have a bunch of relay proteins that are sending a message. The message ends up going into the nucleus. It's going to activate transcription factor, which starts transcribing DNA, which is the product that we want at this point. Some other cellular responses are activating an enzyme, synthesis of a particular enzyme or a protein by activating a gene, so therefore causing transcription and translation to occur. When cyclic AMP, calcium, IP3, or any other second messenger is activated or made, it is generally followed by interacting with a specific enzyme to start the pathway to get the results we want. And so we see a bunch of different response types. Examples of cell signaling in plants, it is very similar, but it is activated by light most often, as opposed to receptor or receiving a hormone. The role of scaffolding proteins. Scaffolding proteins are largely relay proteins to which other relay proteins are attached. A scaffolding protein can increase the signal transduction efficiency. Essentially what happens is, instead of activating one protein every time we kind of have that domino effect, the scaffolding protein holds multiple proteins together so we can activate multiple proteins at once, amplifying the pathway, or amplifying the result that we want. There are videos of this, please look them up. That is a brief overview of cell signaling. What is important is that you apply this to the endocrine system and the nervous system and the muscle contraction, because those are all usable related topics.